Do not touch my experiment. I need to write a lab report on that for my biology IA.
I'm not touching it. What did Campbell just add into the thing? Sodium hydrogen carbonate. What? It basically releases carbon dioxide into the water for the plant. Wait, isn't carbon dioxide what you don't need and breathe out? Yeah, but you're not a plant, or algae, or cyanobacteria. They do photosynthesis. I think I've heard that before. They use carbon dioxide and water to form glucose and oxygen. Glucose is basically their food, and oxygen is released. And that's why there are bubbles. Cool. So they make food and air from air and water. If you want to think about it that way. What's this for? Don't move it! That's for the plant. They need it for photosynthesis. Specifically to split water into hydrogen ions, electrons, and oxygen. This is called photolysis, meaning splitting using light. We told you not to touch it! What's Campbell gonna do with their IA now? Sorry, but the light was too fun. We can try fix it. We don't even know which plant Campbell used. Campbell could be back at any time. You're not helping! I'm thinking we could use chromatography to work out which plant Campbell used. Wait, how do we do that? Follow my instructions. Cut out a piece of the leaf and place it into a mortar. Put some propanone in there. This will dissolve the membrane of the cells. Use a pestle to grind up the leaf. Using a pencil, draw a straight line 1 centimeter above the bottom of a strip of paper. Extract some of the solution and spot it on the line you just drew. Place the strip of paper in the test tube with the solvent, but don't let the drop touch the solvent, or else it will just dissolve before it runs up the paper. Repeat this with all the plants. And now we just have to wait. For how long? It should only take a few minutes. The chromatograms are ready. Let's work out the RF values. The what? The retardation or retention factor. You can calculate it by dividing the distance traveled by the solute by the distance traveled by the solvent. Is that relevant? All these solvents have traveled the same distance. So can't we just compare them by eye? Oh yeah, you're right. Look, this must be Campbell's. Great! Let's set up everything like how it was before. Put some water that has been previously boiled into a beaker. This makes sure that there isn't carbon dioxide in the water. Cut a bit of the plant and put it into the beaker. Add some sodium hydrogen carbonate. And put the lamp where it was. Yay! Everything is like how it was before. Can you explain now what on earth was happening here? I thought all leaves were just green. Leaves have pigments, which are in charge of absorbing light from photosynthesis. There are different types of pigments, all of which absorb slightly different wavelengths. Some absorb red, some absorb blue. The main pigments are chlorophylls, which absorb red and blue light. That's why they appear green. Wait, let me process that. Yeah, the colors you see are the ones that were reflected, meaning they were not absorbed. Oh. Have a look at this graph. These are called absorption spectra. It shows the different wavelengths different pigments absorb. For example, you can see that chlorophyll A mostly absorbs red and blue light, and barely any green light is absorbed by any pigment. You can also look at an action spectrum. Here, we are also looking at different wavelengths, but we are interested in seeing the overall rate of photosynthesis at each wavelength, with all the pigments combined. It looks quite similar to the absorption spectrum, just with all the lines merged together. Yeah, because the more light the leaf absorbs, the higher the rate of photosynthesis. For example, according to these graphs, if I shine red light to the plant, it is going to absorb more light, so it will have a higher rate of photosynthesis. But if I shine green light, not much light is going to be absorbed, so there will be a low rate of photosynthesis.
Hi, I'm back.
Hi.
Great, my leaves are still fresh.
What's your IA about?
I am investigating the role of carbon dioxide concentration on the rate of photosynthesis by adding this and counting the number of oxygen bubbles produced.
That sounds fun. I also want to do an IA. Give me some ideas. You can do a similar experiment, but instead of changing carbon dioxide concentration, you can change light intensity by varying the distance of the lamp to the plant. Any other options? You can also investigate the effect of temperature on photosynthesis by using a fermenter. Inside, there is algae. You can regulate the temperature using a controlled heater and measure the rate of photosynthesis using an oxygen electrode. Something else? I want to do something much bigger. I don't think it's suitable for an IA, but you can do a face. Free air carbon dioxide enrichment experiment. You control the amount of carbon dioxide in an area, but in an open condition. Yeah, I don't think it's suitable for an IA.